In subproject 1, Simon Thorn, our Chinese counterpart Arong Luo, and myself, Heike Feldhaar, will characterize the bottom-up effects of tree diversity on wood decomposition and decomposer interactions. Decomposition is the primary process by which the stores of nutrients and carbon captured by plants are recycled and made available again to plants and organisms of higher trophic levels. Wood decomposition has been shown to strongly depend on environmental factors. As you can see here on the left, it increases with higher temperature and precipitation, with the grayish and red dots indicating an annual mass loss of up to 20 to 60% of dead wood. And while microbes have been considered the primary agents of decomposition, several recent studies, like the one showed here by Seibold and co-workers, have shown that the contribution of insects to wood decomposition also becomes more important towards the tropics. Another important factor driving wood decomposition rate are wood traits. Wood of tree species with a more nutrient acquisitive strategy, here on the right side of the x-axis shown in pink, contains more nutrients and has lower lignin content and lower density. And this softer wood decomposes faster and is also more attractive to insect decomposers, which speeds up wood decomposition again. However, the bottom-up effects of tree diversity on wood decomposition and decomposer interactions have not been studied yet, although multiple factors related to tree diversity can influence the communities of organisms living in dead wood, the so-called saproxylics, and in turn the wood decomposition process. So first, tree diversity can cause environmental changes such as microclimate or soil pH, which may feed back to the decomposer community. Second, tree diversity can directly affect decomposer communities through tree-specific traits, such as the wood traits shown before, or by enhancing the diversity of dead wood and forest heterogeneity. Both of these factors, microclimate and tree traits, have been shown to influence wood decomposition within the BF China experiment before, and we cooperate with Christian Wirth from former projects and will receive the tree trait data from him, especially the wood trait data. On the community level, the increased productivity of more diverse tree communities should benefit decomposer communities as more resources are available. This in turn increases the diversity and abundance of saproxylic insects, but it may also change the biotic interactions among saproxylics and organisms of other trophic levels within and around the dead wood through predation, for example, by ants on termites, that potentially prevent the colonization of dead wood, but also potentially results in higher competition for dead wood objects or facilitation with one organism enabling the colonization of a second. In the project, we plan to characterize and disentangle the influence of all these factors to wood decomposition and the decomposer communities. And now Simon Thorn will give you an overview of the planned work.
In work package number 1, we will survey the natural amounts of deadwood on all experimental plots, which is particularly interesting because the forests are slowly approaching the natural thinning phase. This is coupled to work package number 2, where we will survey saproxylic insect communities on the same plots and on the same deadwood objects. And work package number 3, we will then move on to more experimental approaches, where we will expose artificially deadwood with and without the exclusion of insects. A similar approach will be conducted in work package number 4, where we will also expose artificially dead wood with and without the exclusion of ants at different height levels in trees. The main hypothesis that will be tested in work package number 1 is that amount and diversity of dead wood increase with increasing tree functional diversity. The main mechanisms behind it, which we assume, is that in low diversity plots the deadwood amount are mainly characterized by the dominant tree species, while at high diversity plots the mean amount of deadwood should increase, but at the same time the variance decrease with the increasing tree diversity. Moving on to work package number 2, we assume that with ongoing tree diversity, the diversity of saproxylic insects will also increase. This should be driven mainly by an increasing deadwood amount and diversity, but also by an increasing structural heterogeneity. In work package number 3, we will expose deadwood from different tree species. Those species will be selected to cover very different biochemical traits to test the prediction that decomposition rate depend on those traits and is actually accelerated by saproxylic insects. We assume that softwood, for instance, is more faster decomposed than hardwood, and the effect is particularly strong in the presence of insects. And we assume that the higher insect diversity across the tree diversity gradient will increase the decomposition rate. In the last work package, we will test if the decomposition is faster with contact of deadwood to the ground or in the trees. We will therefore again artificially expose pieces of deadwood and will also apply treatments with the exclusion of ants. We assume that ants will exclude other saproxylic due to composition, and we also again assume that insect occlusion will decelerate the decomposition process. Work package number 4 is a collaborative project to work package number 3 in SP4. This leads to the last slides, where I will briefly introduce the integration of our project into the overall framework. SP1, which is our proposed project, will provide deadwood amount data to SP2 and deadwood arthropod diversity data to SP3. To SP4, we will deliver data on the competition for nesting space in deadwood. We will utilize basic soil data from SP2 and saproxylic community data from pitfalls and flight interception traps from SP3, while we will work in close collaboration to SP6 regarding the ant abundance and trophic niche to determine the predation pressure. Our entire project will be conducted in close collaboration to Arong Luo, who will be in charge of the identification of arthropods and fungi from deadwood using mechaboard coding, and who will have a more deeper look on the co-occurrence patterns of fungi and saproxylic insects. She has planned a German Sino proposal for 2022. With that, I would like to hand